Here's starter. So pause the video, complete it. I'm just about to go over it. Okay, so question 1. Name the 2 types of protein. They are globular and fibrous. Okay, need to know them. 1b, which type can enzymes be classified as? Enzymes a wee bit more complex, so they are globular. Okay, question 2. Now, keyword here, explain what would happen to moist pH paper when a protein is heated with soda lime. Okay, it will turn pH paper purple. Now, the question says explain. It's a keyword, explain. Okay, if the question just said state what would happen to moist pH paper, that's a perfect answer. But because it's explain, not only do you have to state it, you need to explain why it's turning pH paper purple. Okay, so turn pH paper purple as the amine/ammonia gases released dissolve in water to form an alkali. Okay, so just bear that in mind. If the question says explain, make sure you are explaining. Okay, explain why enzymes are specific to one type of reaction. So the active site of the enzyme has a specific shape that only certain substrates, remember that new word? Okay, that only certain substrates can fit. And it's all to do with that lock and key mechanism, remember. Right, 4a, what do we call temperature? Star. Now this is the temperature where the rate's the highest, the enzyme is most active, okay? And we call that the optimum temperature. Okay, what happens to the enzyme above temperature, the optimum temperature, and why? Enzyme denatures. Okay, now why? This is the chemistry behind it. It denatures as I've not explained that yet. That's today's lesson. Denatures as active site changes shape and is irreversible. Okay. C, why does the rate increase as the temperature increases in reactions that do not involve enzymes? That is because reactants contain kinetic energy equal to or greater than the activation energy. Okay, again that's a wee unit 1 reminder. And question 5, what is an enzyme? Arguably probably the easiest question here, however it's always a question that people sometimes just forget. An enzyme simply is a biological catalyst. Okay, a biological catalyst. Right, our heading today, again it's proteins. It will be our last lesson on proteins and it's lesson 3. Okay. Right, our subheading is going to be denaturing. Now I said last lesson I was going to explain from a chemistry point of view what we mean by denaturing. So here goes. Now, wee bit of sort of prior knowledge here. So past the optimum temperature, yeah, and then just put in brackets. We've spoke about this in the starter, but it's where the enzyme is most active. Okay, so past the optimum temperature, enzymes become denatured. Now, we've learned that they become denatured because the active site changes shape. Yeah, it's irreversible, but we need to look at it from a chemistry point of view. Okay, so what I'm going to do, I'm just going to quickly draw a protein. Okay, so I'm putting R because R is just the rest of the molecule. What we're interested in is obviously the N and the C. Okay, so if I just draw a protein, okay, and this protein would be made up of 2 amino acids. Yeah, if we were to break the peptide link, yeah, we'd have an amino acid there, and we'd have an amino acid there. Okay, so in order to show this, I need to draw two proteins. So I'm just going to draw it the same. Draw one after the other. Yeah. Remember, proteins are complex or globular, so they are interwinding with each other, twisting, turning. So here we are, proteins right next to each other, two different proteins, or it could be the same protein, just they've interwinded and they're now next to each other. So what we have, if we think of unit 1, if we think of bonding, yeah, we're going to have a delta positive here and a delta negative here, because carbon's got an electronegativity of 2.5, oxygen 3.5. Okay, so you're going to get that, you're going to get a permanent dipole. Okay, you're also going to get a permanent dipole here and here. Okay, you would with that sort of group there, but I'm just going to focus on these two. Now, what we'll get is you're going to get an interaction. Okay, now what we're interested in here, we're not really interested in the permanent dipole interaction. What we're interested in, because it's stronger, remember it's still weak, but it's stronger in the permanent dipole interaction. But what we would have here, again, we're going to have a permanent dipole because of the electronegativity difference, but because it's a hydrogen covalently bonded to a nitrogen, here we're going to get a hydrogen bond. Okay, so proteins, they contain hydrogen bonds. Enzymes, because they are proteins, they contain hydrogen bonds. Now, what happens? This is a cool part. This is why enzymes denature. So, when heated, the hydrogen bonds again, this works for all proteins, but I'm just going to solely focus on enzymes. So when heated, the hydrogen bonds in enzymes break. Okay, obviously, because we've heated them. Okay, and we've heated them past the optimum temperature. This is when the hydrogen bonds are going to break at the optimum temperature. So what happens once we break them? Once cooled, yeah, and now this is quite cool. Once cooled, the hydrogen bonds, they reform. Okay, they reform, but, big but here. So, but reform in a different place. Okay, remember, proteins are massive, they're polymers. So, There's no chance if we break the hydrogen bonds, are they going to reform in the exact same place? That's just not going to happen. So they reform in a different place. And what that does, that leads to a different active site shape. Okay? And because of that, the enzyme can no longer function. Yeah, it's no longer specific because the active site's changed. Okay, and if the enzyme can no longer function, we classify that as denatured. Okay, so if you get asked about what does it mean if an enzyme becomes denatured, you need to talk about the hydrogen bonds. The hydrogen bonds are breaking, okay? It's not just good enough to say the active site changes shape. You need to explain why the active site changes shape, and it's because the hydrogen bonds are breaking, and then when they're cooling back down, they're reforming, but they're reforming in a different place, okay? So please make sure you are being specific in your answers to that. So, last thing we're going to look at for proteins is all about hydrolysis. Okay, now remember, hydrolysis is the breakup of a large molecule into small molecules using water. Okay, lysis means to break up, hydro means to use water. So, during digestion, enzymes hydrolyze proteins to amino acids. Okay, and the proteins, what we're talking about here, it's the proteins from food. Okay, so say we're eating some meat, what happens, the enzymes during the digestion process, the enzymes break up the proteins and they break them up into amino acids. Okay, now remember, I'm going to make a wee note here. Proteins are large, okay, massive, they're polymers. And amino acids, they're much smaller. Okay, they contain an amine group and they contain a carboxyl group, and that's it really. Now, what happens? Yeah, it's very smart, this. These amino acids, okay, because they're small, yeah, much smaller than the proteins, what happens now, these amino acids are small enough to pass through the gut wall. Okay? Because proteins are too big, they're unable to pass through this gut wall. But if we hydrolyze, if the body hydrolyzes the proteins, makes them into amino acids that are smaller, yeah, these amino acids can pass through the gut wall into the bloodstream. Okay, now this is the part I think is just amazing. So they pass through the gut wall into the bloodstream. The amino acids can now reassemble, okay, into a different order to create new proteins the body actually needs. Okay, so the proteins that we eat from food, they hydrolyze to form amino acids in our bodies, and then amino acids can pass through the gut wall into the bloodstream, and then the amino acids can now reassemble into a completely different order, into a protein that our body actually needs to function. Okay, that's amazing. So what's happening here? Overall reaction is you are taking a protein, you add water, you're going through hydrolysis, and you're making amino acids. Okay, and remember the amino acids can form again to form proteins, and because they can assemble in a different order, it doesn't have to be the same order. That's the cool thing about this. Yeah, it can be a different order into a protein that we actually need. And remember, if we go from if we are joining amino acids together, it's a condensation polymerization reaction. Okay. So let's just show you what's happening here. Now, if I draw a molecule, wait there, let me. So just get everything in. Make sure you copy this exactly. Okay. Right, there's a molecule here. Now, this is what we would call a tripeptide. Yeah, not so much a protein because we've not got the end bonds. It's not an extremely large molecule. It's not a polymer. Okay. So the question is, show what happens when this tripeptide. Now you don't need to know what a peptide is really. Okay, you'd always get explained that in the question. So show what happens when this tripeptide hydrolyzes. Okay, and that word there, tri, that should be an indicator as to how many amino acids this is made up of, but we'll look at that. So state what happens when this tripeptide hydrolyzes, okay? And next part of the question, how many different, okay, this is very important, how many different amino acids make it up. Okay? So, what you need to do is remember when we're hydrolyzing anything that sort of contains the amide link, the peptide link, if whatever you want to call it, is you have to break it up between the C and the N. Okay, so that is you've got an amide link there, and you've got an amide link there. So what happens is you break it up between the C and the N. Okay, and what we would be left with is we would be, if I grab this, we would be left with HNC like that, okay? But remember, because we broke it off here, we're adding a water, the OH would go back along here, and that is our amino acid. So we've got, Mr. H, we've got our amine group, and we've got our carboxyl group, okay? And what else have we drawn? If we've split it here and here, so we've got NCC, and again, we've got a H and a H, and the middle part is just filled with H's, and then the last part is our carboxyl group, okay? Because it's split there, the OH gets added to the carboxyl group, and the H gets added to the NH, okay? So that's a molecule here. Now, if we were to draw this one, okay, hopefully you've noticed that that amino acid would just be the same as the first one. So what happens, we've actually got two of these, okay, but we've only got one of that. So the question, the answer is we've got two different amino acids. We've got 3 in total, okay, and that's where the tripeptide's coming from, but we've got 2 different amino acids because this amino acid here we find there, but we also find there, and there, okay? Right. What we would be doing in class now would be an experiment. And to be honest, I'm really quite glad that we're not doing this experiment because it absolutely stinks. But anyway, experiment. So proteins in a lab can be hydrolyzed. Okay, they can be hydrolyzed back into amino acids, but we knew that, okay, because we've explained it already. Proteins, they can be hydrolyzed and we get our amino acids back, okay. However, it's by a process called refluxing. Okay, this is a new term. Well, I would have mentioned it in class before, but it's the first time we're going to go into detail. So by a process called refluxing. Now, what that looks like, it's quite hard to draw. Is you would have a round bottom flask, okay? I'm going to use a ruler for this actually. And you'd have a tube going up like that. Sorry, it shouldn't be going up that high. But anyway, then what you have is you have something that you'll know what this word, this is. You'll have something here called a condenser. Now, we've spoke about condensers before. We did it in Nat 5. Yeah. A condenser when we looked at fractional distillation, a condenser in a reflux position. And what we mean by reflux is in an upright position. Okay? When you've seen a condenser, it's been at its side at a downwards angle. Okay? However, we're in a reflux position, so we've got the condenser just upright. Yeah? And very importantly, I'm going to use this in a different colour, is if we look at this, the water goes in at the bottom and it goes out at the top. Now that's very important, okay? Water in, water out at the top. Okay, now I've only maybe seen 2 questions like this, multiple choice questions. But you need to know that the water goes in closest to the round bottom flask and it goes out furthest away. Okay, it does make a difference. And obviously we are applying heat to our protein. Okay, now this works in a similar way to how esters would work with the paper towel. Okay, so we heat it under reflux, we heat it with water and it goes up here, turns into a gas, it evaporates. And because we've got water running through the condenser, it's cold here, okay, so it's very warm in the round bottom flask, it's very cold here, so what happens, we heat it up, it evaporates, turns into a gas, and because it's cold here, it condenses, yeah, it goes from a gas back down to a liquid, and we start to get a protein, we start to get our amino acids forming on the top layer, okay? Now you need to know that. Now like I said, we start here, water in is the closest to the round bottom flask, okay? And the water out is furthest from the flask. Okay, so water in, water out. And that's it. That's how we would split up a protein back into amino acids. Right, last thing we need to speak about. Amino acids can be identified by a technique called chromatography. Okay, now you probably already know what chromatography is from like first, second year. However, we go into it in a lot more detail, but we'll look at it at Unit 3. Okay, so see Unit 3 later. Okay, so I'm essentially, we're just going to dip our toe into chromatography here. Now what happens, I'm not going to go into the ins and outs, I'll just briefly summarise it. So chromatography, what happens, chromatography paper is spotted with an unknown amino acid and also with known amino acids. These are then compared. Now this might not make sense yet, but it will when I give you the example. So again, I'm going to use a ruler. Again, just watch how I'm drawing this, and you guys will need to do the same, obviously. Right. So this here is our chromatography paper. This is our solvent. Okay. Now, what happens is we draw a line here with, say, like a pencil or whatever, and then what we do is we spot it with 4, or as many as you want, I suppose. We spot it with amino acids. Now, we spot it with an unknown one, so one that we're trying to work out what it is. Okay, we'll call that X. And then we spot it with known amino acids, in this case 3. So we don't know what X is, we don't know what amino acid X is, but we know what amino acid 1 is, we know what amino acid 2 is, and we know what amino acid 3 is. Okay? So we'll write that down. X is our unknown amino acid, and our 1, 2, and 3, we know what amino acids they are. Now, this is a cool part. So what happens is the solvent travels up through the paper. Yeah, so we dip our chromatography paper in just sort of the bottom part, and then the solvent starts to rise up. Okay, so it travels up through the paper, and it takes the amino acids with it. Okay, each amino acid travels a different distance. Okay, and what I mean by that is that, let's just say X, the unknown one, that travels up to here. Okay, amino acid 1 might just travel, say, to here. Amino acid 2 might travel up here. And amino acid 3 might travel here. Okay, so what we can do, we can look at X, we can look at the one that we don't know what it is, yeah, and we can compare it to 1, 2 and 3, and hopefully you can see that amino acid X and amino acid 2 have travelled up the same distance. Okay, now because they've travelled up the same distance, what that tells us, amino acid X and amino acid 2 are the same amino acid. Okay, so that's why we do chromatography, because we can have unknown samples, compare it to samples that we know about, and then that will tell us what the unknown sample is. Okay, now, and I'm not going to explain why that happens, I'll explain it when we actually do chromatography, but for now, just accept that because X and 2 have travelled the same distance, they're the same amino acid. Okay, so let's write this. So from this, it can be seen that the unknown amino acid found in our hydrolyzed protein is amino acid 2. Okay, so again, this sort of links in. So what we've done here, we've started with our experiment, we've took our protein, we've hydrolyzed it, and we've got different amino acids. We've taken one of those amino acids that we don't know, we've used chromatography, compared it with amino acids that we do know, and we've worked out that amino acid X and amino acid 2, because they've travelled the exact same distance, they're the same amino acid. Okay, and that's how chromatography works. Right, so let's summarise what we've learned. So at the start of the lesson, we spoke about denaturing. Okay, so that's where we are going beyond the optimum temperature, and what happens, not the active site, of course, is changing shape, but we're interested in why that active site is changing shape. It's changing shape because the hydrogen bonds between the NH and the NH, they're breaking, okay? And then when they cool back down, they're reforming, but in a different position, okay? And if they're in a different position, the shape's different, okay? And that's what we mean by denaturing. So, then we went on to look at hydrolysis. Hydrolysis is when we sort of add water to a protein and we hydrolyze it to form amino acids. Okay, and we spoke about how that happens in food. So the protein from our food hydrolyzes in our bodies, and those amino acids are small enough to pass through the gut wall into our bloodstream, and then amino acids reassemble in a different order to create a protein that our body actually needs, which is pretty impressive actually. Okay, now remember, if we're hydrolyzing a protein, it's breaking between the C and the N of the amide link. Okay, the C and the N of the amide link. Very much like how esters break between the C and the O of the ester link, proteins break between the C and the N of the amide link. Okay, and that can tell you the different amino acids that you've got. Just remember when you're drawing amino acids, you're adding the OH onto the C double bond O, and you're adding the H onto the N. Okay, because if you draw it without that, that's not an amino acid, you wouldn't get the mark. Okay, then we went on to hydrolysis as an experiment. Just remember we've got a condenser in a reflux position, upright, water in at the bottom, water out at the top. Okay, and we're heating it up, and it just acts as a condenser, yeah, evaporates, cools round about the condenser, and then it falls back down and it forms an amino acid. Yeah, and then finally we can use chromatography to work out what amino acids we have. Yeah, so I'll let you look through that because I've just explained all that. Right, that's us for today, so thank you.